out. And so, yeah, it's really, I don't know of another example in evolutionary theory of a prediction that clean of something that obscure.
I know one.
Oh, yeah?
Yeah. I once heard a story about a graduate student who predicted that the breeding protocols of laboratory rodents would compromise the laboratory system in terms of its relationship to so-called wild type versions of the same species. So you have the bred rodents and you have the wild rodents, and that they would be distinguished by virtue of the fact that the non-coding nucleotide sequence at the end of the chromosome, known as telomeres, would be wildly different in length if the prediction were true from pure evolutionary theory.
Wow.
Yeah.
Yeah. Yeah, that story, it didn't happen exactly the way you said it, but, you know, it's been a lot of years and it takes a second to get back there.
Yeah, I mean, it's you did that.
Yeah, I did that.
And that story, unfortunately, has not really been told. And it is, in some sense, your central origin story as a biologist.
It's a pretty good one, and it definitely changed the way I saw myself in a way that has been very productive.
Okay. I want you to talk to me about that story, and I will, because I lived it with you. I know that it happened, and I know that it got buried.
Yep.
And I know that it's part of what I'm calling the distributed idea suppression complex, because quite frankly, you were not the only person who was a part of this story, and the story had to die because it said something, which is that the power of your theory was sufficient to predict from first principles a manifestly observed and surprising result within molecular biology from pure evolutionary principle.
Yep. All right. I'll try to do a short version of it.
You know, this is long form podcasting, and you tell however long the story is, I guarantee you when people finally figure out that it may be that the rodents that we've used to test drugs on, let's say, might be compromised in a way that would be potentially extra permitting of potential toxins in the form of pharmaceuticals. I think that it's going to be fascinating. It's going to repay the study that it will take to understand the story. The floor is yours.
All right. So let me just set the stage a little bit. Evolutionary biology
But do me a favor.
Yeah.
You can get into a very patient, careful, pedagogical mode. This is an exciting story. Tell it the way it actually occurred.
I'm going to tell it the way it actually occurred. And I'm going to be careful. I'm going to try not to be... there are parts of it that were for a very long time kind of emotionally fraught. But anyway, I think I remember it well enough to do a sparse but complete version.
Okay.
Evolutionary biology has long been biased in the direction of abstraction rather than thinking about mechanism. That is to say, we deal in the phenomenology of things. We talk about gross patterns that we see in nature rather than talking about the fine detail of what drives them. That has been changing in recent decades, but it has a long history, and it comes from a very mundane place. That mundane place is that we just haven't had the tools to look, for example, inside of cells, and we haven't been able to read genomes. You know, we've been able to read a gene here and there at great expense, but the ability to peer into genomes is pretty new. The ability to peer into these molecular pathways is pretty new. So anyway, there's a historical bias in evolutionary biology against mechanism and in the direction of phenomenology. I have never been particularly fond of that bias. I've always been interested in mechanism. I'm interested in the phenomenology too, but I've always kept my foot in the door with respect to mechanism. And as a undergraduate, I took lots of mechanism classes. I took a development class. At the time, developmental biology was, in my opinion, a bit stuck. It is now unstuck in a very dramatic way. But anyway, I took a developmental biology class. I took some immunology or immunobiology. And anyway, I was armed with these things in an environment in evolutionary biology where most people were not. Most people were in the phenomenology. And one day I happened to be in a seminar. Dick Alexander was running a seminar for graduate students, and a student was there who was very out of place. He was studying cancer, and he, on a lark, decided to take an evolution seminar that looked good to him in the catalog. And it wasn't right for him. And he gave a talk at some point. And his talk was on his work with cancer. And frankly, because all the other people in the room were evolutionarily oriented, nobody was really tracking what he was saying. But what he said struck me like a bolt of lightning. He said that in the realm of cancer research, people were looking at telomeres, which are these repetitive sequences at the ends of chromosomes, and they were toying with the possibility that the fact that these telomeres shorten every time a cell divides, that is providing a resistance to tumor formation. Very straightforward. Counter counts down, and that would prevent.
So let's just, for the audience that maybe needs a tiny refresher, we are taught in general that DNA is a string of letters called nucleotides, A, C, T, and G, and that in general, 3 of those that are adjacent to each other form words called codons, and for every word, there is an amino acid or an instruction to stop coding for amino acids. So this is the instruction tape that tells us how to string together amino acids into proteins to make machines, molecular machines. This is some weird, different thing where the region of DNA could be interpreted as coding for a protein, but in fact might be instead just counting how many nucleotides are at the end. So it comes across as a counter.
It's a little better. It was known not to be a coding sequence. It wasn't a useful sequence. Okay. So what you had is a bunch of DNA at the ends of chromosomes that's just repetitive. And the length of the number of repeats varies, and the number of repeats correlates with basically how many times the cell can divide before it refuses. This being interpreted as a cancer prevention thing made perfect sense. But the reason it struck me like a bolt of lightning was that I was well aware of the existence of tumors and their implication in something entirely different. What they had been implicated in, as far as I was aware, was something called Hayflick limits, which were the tendency of perfectly healthy, happy cells to grow and grow and grow and grow in a petri dish until they hit some number of divisions and then to stop without apparent dysfunction. So
this was the theory of Leonard Hayflick.
Yep. It was the discovery of Leonard Hayflick, who basically overturned the prior wisdom about cells, which was that they would grow indefinitely as long as you kept feeding them and making an environment that was conducive to division. So I don't exactly know why that result had been misunderstood at first. Maybe somebody had a cancerous cell line, and so they got the wrong idea and it just spread. But Hayflick checked it, and it turned out to be false. It turned out there was a number of cell divisions that healthy cells would go through, and then they'd stop. The mechanism was not obvious to Hayflick, but later it became clearer and clearer that the mechanism was these sequences at the ends of chromosomes, which shorten each time the cell divides. And the implication was that potentially this was a cause of what we call senescence, what in common parlance would often be called aging, the tendency to grow feeble and inefficient with age. If your cells are each in a cell line, and that line has a fixed number of times that it can replace itself before it has to stop, then at some point your repair program starts to fail. And that repair program failing across the body looks like what you would expect aging. Aging follows the pattern you would expect if cell lines one by one stopped being able to replace themselves. So
we know that there's a special sort of, I don't want to call it cell line because you keep correcting me for every tiny mistake I make in speech. But if we divide our body into 2 kinds of cells, soma and germ, where germ lines are that which has a hope of immortality through reproduction, then it's the somatic cells that have finite limits on their ability to undergo mitosis and cellular repair and whatnot.
Yep. And the germline can't, because if it did, your lineage would go extinct as a result of
small addendum.
Small addendum. So it's the soma, the parts of your body that don't go on to produce babies, that have this effect. The reason it struck me like a bolt of lightning was that I was aware of another very elegant piece of research done by a guy named George Williams. George Williams had finally...
One of the greatest of modern evolutionary.
One of the greatest modern evolutionary biologists. I actually knew him a bit too. He is also now gone, unfortunately. But George Williams had laid out in a beautifully elegant paper the evolutionary theory of senescence. It is an absolutely elegant argument that says that in a lifetime, let's start somewhere else. A creature is built of parts and traits. It has a relatively small genome and a relatively high complexity. At the time, it was thought there might be 100,000 genes or something, and you have maybe 30 trillion cells with a ton of complexity. In order to get that small number of genes to dictate how to produce a creature that complex, the genes are doing multiple things. Williams' point was, when a gene has multiple effects, what we call a pleiotropy, those effects may be good or bad. If effects are good early in life...
By good, we mean contributing to fitness.
Fitness-enhancing traits. At some cost late in life, then they will tend to be accumulated by selection. And the reason for that is because, well, there are two ways to think of it, really. If a negative trait occurs very late in life, then a large number of individuals who have the gene for that trait will not live long enough to experience the harm. So if it came bound to a positive thing early in life and you're dead before the late life harm accrues, you got away with it, right? So Williams' point was, and he was building on earlier work of Medawar, but let's skip that for the moment. His point was, because of trade-offs, you will have lots of traits that are good early and bad late. Selection sees the early traits much more clearly than it sees the late traits, and it prioritizes them because of the discounting that arises because so many individuals aren't around to experience the late life harm, and if they are around to experience the late life harm, a lot of their reproduction is behind them anyway. So they count less. Selection counts more early in life, and this timer starts at the moment of first reproduction, the usual moment of first reproduction
viability
for your species. Yeah. So this is a beautiful hypothesis, and it was beautifully articulated with many predictions, which is the way really good work is done. And we knew at the point that I was entering graduate school, we knew that the hypothesis was right. It was a theory. And the reason that we knew it was right...
The hypothesis is the antagonistic pleiotropy hypothesis.
Yeah, antagonistic pleiotropy hypothesis for senescence. We knew that it was right because it predicted so many phenomena in nature that we could readily go out and measure. And this is, again, where the phenomenology versus mechanism comes in.
Okay.
We know that creatures that are poisonous or have a shell that protects them or can fly away from danger are disproportionately long-lived for their size. Small creatures tend to live shorter lives than large creatures, but if you can fly, then you're off the line of the other creatures of your size. So, for example, there are small bats who have been recovered after 30 years in the wild. So creatures that have special protections have disproportionate longevity. This matches Williams' hypothesis because it is their ability to fly away from danger that makes the likelihood of their experiencing late life costs go up.
Yep.
So selection sees their late life more easily than it sees a small creature.
Look, I just want to say something.
Yeah.
This is a podcast. It's an unusual podcast, and we can talk science, and I'm thrilled. But we always have our colleagues in our minds when we're talking to a general audience, and the colleagues are always in a gotcha mode. Well, you forgot about this, and you didn't mention that. I'm even interjecting little bits because I want to make sure that you're immunized from all the bullshit that the academic. So I just want to make a general statement, which is we can come back and get into any level of specificity that somebody wants to if they want to take you down. I don't care. What I'd love to do is to tell the story with enough punch that people understand what happened.
So we're about to jump into the
meat
of the matter. The theory of antagonistic pleiotropy was well established. But in 4 decades of research on the genome, nobody had found a gene that matched it. So that we knew that this explanation was right, but we couldn't find the genes that caused it. The mechanism was missing. So anyway.
For something to be a gene, it has to be protein coding?
Yeah. Anyway, I knew this as sort of, I was well familiar with Williams' paper at the point that I saw this talk on cancer, and I knew already about the question of senescence. Everything came together. This was obviously the answer, where the missing pleiotropy was. Well, the missing pleiotropy had to do with a telomere, which wasn't exactly a gene. It was genetic. It was DNA, but it wasn't a gene. But it was perfectly capable of producing exactly the effects that we see in senescence across the body. Tissue
So a counter,
after tissue.
not a protein, could be the answer.
Right. Now, I saw this instantly at the point I heard this talk. I raised my hand and I tried to articulate what was so obvious in that moment. And I couldn't compel a single person in the room. They couldn't even understand what I was trying to say. And that,
which is bizarre.
It was bizarre. I mean, Dick was in the room, and, you know, Dick was very broad-minded, and I just couldn't make it clear.
Okay. Let me just interject something. You can correct me if I'm wrong. But my impression of it is that it was a very simple idea attended to by an outrageous amount of irrelevant complexity that had to be very carefully pried off of the central idea.
Yeah, I think that's well said. So anyway, I left the room feeling like I had just glimpsed something so important. And, you know, I wondered, could it be right? And I started to just do the first bit of library research to figure out whether somebody else knew what I knew or...
So I'm not even sure that you fully said it. I want to make sure that I'm even clear on it. And I think I'm right, but correct me if I'm wrong. What you're saying is, what if the Hayflick limit is a protection against dying from immortality at a cytological level? That some cell gets a dream of immortality that it shouldn't have because, let's say, it's a somatic cell. And it says, okay, I just want to keep dividing and dividing and dividing. Nature knows how to do this. And that immortality, which sounds good at first, is actually called cancer.
Yeah.
And so in computer science, we would say, okay, you've introduced a recursion limit into a while loop or a for loop to make sure that you don't have a resource leak, which is what a tumor is.
Yeah. So let me say it this way. If you have a damage to a tissue, cut on your arm or something, cells on both sides of that cut suddenly become aware that there is a problem, a gap, because they can't hear a neighbor on one side of them. And their natural reaction is to start growing into the gap until they can hear a neighbor, which is the sign to stop. If you imagine that something like that is occurring in every tissue, or almost every tissue, the problem is that means that every tissue in your body for which that story is about right is in danger of having damage from radiation or whatever turn it deaf to its neighbors. A single cell that has turned deaf to its neighbors will suddenly start replicating. And if it is deaf to its neighbors, then there's no message that it's going to hear that's going to tell it to stop. So that thing, imagine any cell in your body just taking off and growing and growing and growing.
Okay, so this is terrifying. What you're saying to me is that if I'm comprised of, let's say, 30 trillion cells, and I view them as each, let's say, subroutines, any subroutine that is not denucleated, right? Like this wouldn't happen in the lens of your eye because the nucleus has been removed. But any other reasonable cell is potentially your assassin because its mitosis process might completely go rogue.
It can run away.
Okay.
And so the rather elegant and very simple idea is that there would be a hard limit so that any cell that had become damaged, so it started down this path, would just simply run into the number of cell divisions it was allowed in a lifetime, and it would stop.
So like the moles on my face that some of my less couth commenters love to talk about.
Yep.
Are effectively attempts to kill me that may have stopped, and that the perimeter where they stop is where the Hayflick limit took over and said this cell line must die so that the patient will live?
Yeah. The name I gave them was prototumor. Right. And the idea is a prototumor is a patch of cells arrested at their Hayflick limit because they had become unregulated. If you go to the dermatologist and you say, what do I look for? They tell you certain things to look for. So a round patch of cells that suddenly becomes irregular in shape, well, that's what would happen if you took one of those cells and gave it a second mutation and it started growing again.
Got it.
Right? So anyway, the idea that a limit on cellular reproduction is adaptive to protect you from cancer.
Okay, so there's a little bit of a mind bender, because what you're telling me is that I've got to avoid immortality, which can kill me, and that the solution to not dying is death.
Yes, and that what selection does is it balances these two competing forces to give you as much vigor and longevity as it can.
So all of the other diseases and insults and things that I can die from sort of start to fade away. And at the complete core of biology in this theory, there are two things that I can't get away from. One of which is death by immortality, and the other one is death by recursion limit.
Mm-hmm. That's it.
It's a very elegant thing. And now the problem is that there's all this weird attendant complexity that you had to deal with. So there's like stem cells versus germ versus...
So when I went into the literature, what I found was that people had played around in the neighborhood, but that there was a particular fact which blocked every attempt to make sense of what was going on. And the fact was that rodents were understood to have ultra-long hypervariable telomeres. And I didn't know what that meant at first, but the more I looked into this possibility, the more I realized that dozens of long-standing problems would be solved if my hypothesis was true, but that my hypothesis couldn't be true because basically mice have long telomeres and short lives. Why is that? And I banged my head on the table for a couple weeks trying to figure out what was going on. Yes, maybe even literally on occasion. But the question was, I began to wonder if there was something wrong with the idea that mice had long telomeres. Sometimes, like in Hayflick's case, it turned out that a bunch of people were copying some wrong result in it, so it seemed like a lot of people had seen it, but only one had. And I checked. Was it true that there was that everybody was parroting one study that said mice had long telomeres? And it turns out lots of people had tested it. Mice have long telomeres, like 10 times the length of human telomeres. It just didn't fit. So finally, it occurred to me that it was possible that what was going on, I discovered something in trying to figure out what they meant by mice, right? There's a lot of species of mice, but all the mice that we use in the lab, with rare exception, are from one genus and often from a particular target species.
You were focused, if I recall correctly, on Mus spiritus?
No,
Mus musculus?
Mus musculus, which is the common one.
Yeah.
What shocked me was that it turned out all the Mus musculus that were being used in labs across the country, and in many cases farther afield than that, were coming from one place, which I had no idea. There was one...
I remember getting a phone call when you said, what do you know about the Jack's lab?
The Jack's lab in Bar Harbor, Maine.